All right, welcome back, Year 12 students. So we're just going to run through our second PowerPoint here. So quick recap of what we did in the first video. We looked at nucleotides. So the structure, as you can see, we have the cartoon version here and the chemical version here. All right, we also looked at the actual specific atoms and how all nucleotides essentially have 5 carbons. All right, or a pentose sugar, and that either has this particular oxygen removed to make it deoxyribose or present to make it ribose. Okay, so DNA and RNA, which we're going to look at more today. All right, the other basis is pretty much T gets replaced by U. All right, so A and U go together just like A and T go together. And that's only if it's RNA. Cool. So we're going to look a little bit more at DNA and RNA today. Again, that's your dot points. Let's continue on. Cool. So DNA consists of 2 strands of nucleotides bonded together, whereas RNA is this one or single strand. Okay. And the DNA, it runs in an anti-parallel fashion. So what does that mean? Anti-parallel. Each strand, well, the second strand is upside down compared to the first strand, or each strand is a different direction. Cool. All right, so in nuclear DNA, the helix structure also coils around what we call histones, and they're these proteins here shown in the, I guess, the blue circles with the cross. All right, and that has your DNA strand running around it a few times, and then it continues on to the next one. Okay, that also contains epigenetic markers and other things. All right, and that coils up together to make this chromatin. And that chromatin then coils up into what we know as our X-shaped chromosomes. All right, DNA, this is showing the anti-parallel nature. That's an important word. Okay, so you need to be aware of that. So we have our nucleotides here. All right, individual nucleotide. So if I draw on this, individual nucleotide, boom. All right, so you can see your pentose sugar, phosphate group, and your nitrogenous base, in this case, cytosine. All right, and that's connected through these bonds. If I just delete this. Okay, that's connected through the 3 bonds you see here, all right, to the guanine. And the guanine is shown being upside down, so that G is upside down. The 3' end is upside down, and the arrow is also pointing the opposite direction. Okay, so that's showing how it's anti-parallel. In this nucleotide's case, you have the T, thymine, connecting with the adenine here. As you can see, there's 2 bonds, not 3. So I have a weaker bond together than the C and the G, which I alluded to last video. Just looking at RNA and DNA together, okay, so we know that DNA is our double-stranded nucleic acid, and our RNA is our single-stranded nucleic acid. They both have nitrogenous bases, they both have a sugar phosphate backbone, also known as the pentose sugar, as well as the phosphate group that I just mentioned. And the only other difference, as mentioned in the previous video, is uracil will be within RNA, replacing thymine. Okay, so again, C and G, cytosine, guanine, and then A and T, adenine and thymine for DNA, and then C and G are the same for RNA, and then adenine and uracil, A and U for RNA. Okay, and you might think, that's a singular strand, so when would that actually need to pair up? And we'll get to that later when we learn about transcription. Alrighty, so RNA, single strand of nucleotides, and the picture here shows a particular biology joke that has to do with RNA. Okay, so if you understand it, tell me what it is in the comments below. All right, so RNA is used in many different things. It could be tRNA, so transfer RNA. It could be mRNA, messenger RNA, or it could be rRNA. Okay, so ribosomal RNA. And they're all found doing different things. Okay, main component for coding in protein synthesis as well. So RNA is really what proteins are made of. So the DNA gets split, RNA gets coded, etc., etc. And then translation occurs, and bang, you get your protein. But we'll learn about that in another video. Alrighty, we have our types of RNA. Okay, so again, ribosomal RNA is the rRNA. Okay, and you can see the ribosome here. So you have this this double structure, okay, and you can see that's the RNA running through it, this green ribbon substance, the RNA running through the other part, and then obviously there's a bunch of amino acids there as well, right, making up that protein, as well as rRNA, which makes up your ribosome. Cool. You also have your transfer RNA, so tRNA, and that is a dead giveaway, the tRNA, because it is in the shape of a T, okay. And yes, this looks complex. You don't need to know the anatomy of the tRNA except for the anticodon part. You do want to be aware of that. Okay, so that anticodon, as you can see, will have its base pairs. So C and G, U and A, C and G. They are the complementary base pairs that code on to one another. Okay, and they will attach there, and there will be, in theory, an amino acid up the top here. All right, that gets pumped out of the ribosome, but again, that's another class for another day. Okay, and that there is the process of translation. Cool, so you have your rRNA and tRNA. All right, and then lastly, mRNA, so messenger RNA. You might also hear it referred to as pre-messenger RNA or post-messenger RNA, and that when we look at the process of transcription and translation, we will get into that as well. Okay, so messenger RNA formed in the nucleus, created by the process of transcription, and essentially it's a copy of some sort of DNA code. Okay, obviously it's a half a copy as well, so keep that in mind. So it carries a nucleotide sequence, so generally that's a gene of some description or some sort of coding. All right, and it's messenger RNA is made by a specific protein, which is an enzyme. All right, and how do we know it's an enzyme? It's ending with A's. Okay, anything ending with A's is an enzyme, and that one's RNA polymerase. So that creates the messenger RNA. Cool, and it will look like that, as we know, the single strand structure. Cool, so just recapping that off to finish the video up. We have mRNA, which is your messenger RNA, carries a copy of the genetic code from DNA in the nucleus to the ribosome. We have transfer RNA, which is that T shape, so tRNA, and it carries this amino acid here from the cell cytoplasm to the ribosome, which then in turn will get connected to the polypeptide. Okay. And lastly, we have the ribosomal RNA. As you can see, it runs through the ribosome. It comprises 60% of the structure of ribosomes. The other 40% is protein, so amino acid sequences, polypeptides. Okay? So mRNA, tRNA, rRNA. Beautiful. Thanks for listening. I'll see you in the next video.